All right, well, we are going to get started. This is very exciting. A hybrid grand rounds. We've got a lovely showing in person and many people online. So I'm delighted to have you all in person as well as online. And today is a very special medical grand rounds. It is the Bill Bussey Endowed Lectureship. We are very delighted to have Dr. Tina Hartert as our lecturer this year, our visiting professor. Dr. Jajor will introduce her and Bill Bussey in a sec, but I will say that I first met Tina probably 25 years ago now. We served on the ATS membership committee way back when, it was a lot of fun being on the committee with Tina and getting to know her. And I've been following her career ever since then. And it's a delight to be able to welcome her to Wisconsin. So with that, I'm going to hand it over to Dr. Jajor.
Good morning. It's my pleasure today to introduce our William Bussey Department of Medicine Grand Rounds. Dr. Bussey is very well known to all of you. He is a Wisconsinite through and through, went to undergrad, medical school, did his residency as well as fellowship here at the University of Wisconsin, completing that in 1973. Joining the faculty in 74, rising through the rank to a full professor in 1984. And currently he serves as an emeritus professor of medicine. Over his career, Dr. Bussey has done, practically speaking, everything that you consider important in the specialty here in Wisconsin, in the United States, and across the globe. He served as a section chief for allergy from 78 to 2004, department chair 2005 to 2009, the chair of the American Board of Allergy Immunology, 1995, the president of the Quad AI in 2000. He chaired the United States Asthma Guidelines from 2003 to 2007. The American Thoracic Society bestows an award on one person who has the most impact on the specialty. And in 2014, Dr. Bussey was the recipient of that award, the Breathing for Life Award. Dr. Bussey has received probably the most NIH grant in this institution ever. He had the largest award in the history of the university, the Inner City Asthma Consortium, and this has been going on for, I think, 4 cycles now. That's the last cycle, what that bill was, the PI in 2014 to 2021 was $70 million. It has continued here in Wisconsin in his tradition of supporting others and mentoring. It has been passed on to the next generation. Today, it's my honor and privilege to introduce to you one of our dear friend and colleagues who is going to be our 2022 William Bussey Visiting Professor of Medicine, Dr. Tina Hartert. Dr. Hartert is a professor of medicine and pediatrics and the Lulu Owen Endowed Chair at Vanderbilt University, where she also served as the Vice President for Translational Science and the Director for their asthma research program. Dr. Hartert received her bachelor's degree with honors from Brown University, her medical school with a degree and MPH from Vanderbilt, followed by medicine residency and chief residency at Hopkins, returning to Vanderbilt to do her pulmonary critical care fellowship and remaining there on the faculty ever since. Dr. Hartert has been elected to a number of scientific and medical honor society, including the Alpha Omega Alpha, the American Society of Clinical Investigation, and the Association of American Physicians. Tina has received also a number of awards for her teaching and mentoring. She's so valuable for her mentees, student, resident, and junior faculty alike. And she was the inaugural recipient of their Research Mentoring Award in Translational Sciences. Tina has also received a number of grants from the NIH. There are, 3-4 pages of NIH grant on her CV. But these NIH grants were not just there sitting idles. These have led to a major contribution to our understanding of asthma and allergic diseases, with more than 250 publications to date. And she keeps on going with many publications every single year. Tina remains a very active clinician. She takes care of patients in the clinic as well as on their inpatient service. Dr. Hartert is an internationally recognized researcher in asthma and allergic disease with a special focus on the role of respiratory tract infection in asthma. This is an area of special interest that we share with Tina at the University of Wisconsin. Therefore, it's so appropriate and so wonderful that Dr. Hartert will speak to us today on asthma, the story of an infectious etiology of a chronic disease. Please help me welcome Dr. Hartert to the podium. In honor of this lectureship, we would like to present her with a plaque shaped appropriately in the shape of Wisconsin. Tina Hartert, MD, MPH, William Bussey Visiting Professor of Medicine, 2022. Tina?
Thank you so much, Nisha. Thank you.
I'm allowed to take this off, huh?
Yes.
Great. Can you all hear me? I want to make sure I'm turned on. Great, thank you. Well, it truly is a pleasure to be here today and to be invited to be the Bill Bussey visiting lectureship is really truly an honor. I'm a respiratory epidemiologist, and I used to have to explain to people all the time what that is. And now with COVID, I feel like all these terms that we use are just in the common vernacular. So I'll skip that part since I think you now all understand what a respiratory epidemiologist does. But my entire career, I've been interested in the developmental origins of asthma. And what I'm going to talk to you today about is a story of the long-term consequences of a respiratory virus called respiratory syncytial virus. And the long-term consequences of respiratory viruses is of germane interest and has become a much more common interest with COVID. So hopefully you'll enjoy this story today. These are my disclosures. I have several disclosures that on DSMBs and consultants, but this is completely unrelated to my talk today. And I'll also disclose my grant funding for the work I'm about to present, which is from the NIH, the World Health Organization, and the Bill and Melinda Gates Foundation. So I want to also make just a few comments about Bill Bussey, who truly embodies what we call the triple threat as a mentor, a scientist, a role model, and a colleague to many of us. Very early in my career, when I was finishing my fellowship in the late 1990s in junior faculty, I actually first met Bill. And Bill otherwise didn't know me from Adam. I wasn't at his institution. I'm not an allergist. But from the very first meeting of Bill, he reached out and was an incredible proponent and sponsor of me as a junior faculty member and authored a paper together, which coming out of fellowship and having Bill Bussey co-author a paper with you is just enormous. He has built here at Wisconsin what is really one of the premier, probably the premier asthma research program in the world. And at the time building this program, it was really this novel clinical translational model, not siloed in a single department, but across medicine and pediatrics and basic sciences to understand asthma and asthma development. And I really learned several fundamental things from Bill. And I was just joking with him because my husband also studies asthma. He's a mouse doctor. But I come home and Bill, I don't know if it's biweekly or whenever, but in our house, because Bill has conference calls at 8 o'clock at night, I hear Bill's voice booming through our whole house about every other week or so. But I've learned several very fundamental things from Bill. as well. And I love these two pictures in particular because even though I don't get to interact with Bill frequently, I've come to learn that when Bill kind of leans forward and reaches out like this is the key pearls that you're supposed to about to take away. Although I really would love to know what the badger here was learning. There were a few things. First of all, his generosity of sponsorship and mentorship. And I think that's so important for us as scientists to give back to the next generation. And I was certainly a recipient of that. And for that, I'm really very grateful. And modeling this for the next generation. You know, the other thing that he did that I was really struck by early on was his sponsorship and support of women in science, which at the time, having male colleagues promote those who are underrepresented in science was really rare. And I've always remembered that and tried to do the same. And then lastly, you know, I've seen that he's recognized the importance of having a larger vision, not just for the research question that we're answering, but actually how we do our work as well. And the model of our science and the camaraderie that we have with our colleagues is so tremendously important for us enjoying our career and what enriches, I think, so many of our careers. And I felt incredibly fortunate over the years to be able to collaborate with the group here, Jim Gurn and Rob Lemansky and Dan Jackson and Chris Saruji and Anne-Marie Singh. It's just been absolutely tremendous what has been built here and the ability to collaborate with this group, for which I'm really grateful. So Bill, I'm really, truly incredibly honored to be able to give this lecture. And I'm grateful also more than 2 years into this pandemic, actually, to be here in person, to be able to meet with all of you. And if I get really glitchy during this talk, it's because I haven't given one of these, not in my office behind a Zoom screen in 2 years. So be kind to me, please. So this is a very brief overview of what I'm going to talk about today. I have a single slide about asthma. I'm going to talk about the exposure, respiratory syncytial virus, which we all call RSV, and I'm going to call RSV through most of the talk. An outline of the historic problem of what our approach to understanding the relationship of RSV and asthma is, because I think it's really germane to the studies that we are doing going forward. The multi-level evidence for explaining how a virus might contribute to later chronic respiratory disease. And I'll end with conclusions and the implications. So these are really our motivating questions. As I mentioned, we're interested in the developmental origins of asthma. And we are interested in a multitude of different environmental exposures. But today I'm going to talk about one, as I mentioned, respiratory syncytial virus. So our really motivating questions are, does RSV infection increase the risk of asthma? Does it cause asthma? And if it does, how does it cause asthma? Because ultimately what we really want to answer here is, would preventing early life RSV infection actually prevent asthma development? So very brief background about asthma, just to ground us in what the global health implications of this disease has and how we need to understand it and trying to understand its developmental origins. A number of years ago, the World Health Organization deemed asthma one of the most significant non-communicable diseases worldwide. 358 million people are estimated to have asthma, so it's one of our most common chronic diseases. Asthma, we know, is highly heritable. And although we've identified over 110 or 20 or so genes, even small combinations of those genes haven't led to or predicted disease development. We all know in the clinical setting, if we just take a family history of asthma, it's highly predictive of whether that child will go on to develop asthma in later life. But we also know that asthma is a disease that results from gene-environment interactions. And this is really important because what it means is that we can potentially modify environment and modify the risk of developing disease. And that really underlines what I'm going to spend most of the time talking about today. And then why, as an adult pulmonologist, do we study children? And the reason is that 80% of the onset of this disease is in childhood. So the place to focus in preventing this disease is during pregnancy and early life. So I'm going to convince you first, I think, by way of setting that asthma is potentially a preventable disease, because I think this is very important in understanding its developmental origins. So why should we believe that it's actually a preventable disease? Well, there's been a marked increase in rates over decades. And although since about 2010, we've seen a slow decrease, these marked changes over relatively short periods of time can't be attributed to genetics and really have to be driven largely by environment. We also know that there are marked differences in rates of asthma across the world. Here in red, these are the countries with the highest asthma prevalences in the world, in the yellow, the lowest. And even in contiguous countries like the US and Mexico, there are marked differences in asthma prevalence. But there are also marked differences in asthma prevalence in genetically homogeneous populations that live right next to each other. And East and West Germany is probably one of the best examples of this, where in West Germany, asthma prevalence was 33% higher than in East Germany. In East Germany, asthma prevalence was really very low, even compared to most of the world, about 4%, and increased following unification, to suggest over this really short period of time, these genetically homogeneous populations had marked differences in asthma rates, which really, again, can only be attributed likely to environment. And then lastly, we've seen changes in asthma prevalence with relocation. So in the US, Puerto Rico has one of the highest asthma prevalences, in fact, one of the highest asthma prevalences in the world. But when Puerto Ricans relocate to the mainland US, subsequent generations have very significantly lower asthma prevalence. And so in sum, what this tells us is that asthma has marked differences in contiguous populations, even those who are genetically homogeneous. We've seen marked changes over short periods of time and marked changes with relocation. And what this tells us is that asthma then is not the end-all be-all genetically programmed disease that will develop if you have a family history of disease, but that it's potentially modifiable by environment. And from a public health perspective, environment that doesn't take really expensive drugs or fancy medical infrastructures. It means modifying environment, diet, lifestyle factors, and we can modify development of disease. So then I'm going to tell you just briefly about the exposure that we're going to talk about today, respiratory syncytial virus. So this is a respiratory virus that was discovered in 1956 by Robert Chanock. It's a single-stranded RNA human orthomyxovirus in the same family as a similar virus to measles. And it's a small virus. You can see here on the right side, there are only 11 genes which encode for 10 proteins in this virus. And important are the surface glycoproteins F and G, which, like the spike protein for SARS-CoV-2, which the whole world has learned about, these are the surface glycoproteins that are very important. I'm going to talk a little bit about them later in the human immune recognition of this virus. This is a highly infectious respiratory virus. It infects everyone by the time we're aged 2 to 3 years. And infants and older adults suffer the greatest morbidity from this virus. And the R naught for this virus, if you don't have prior immunity, which is going to be infants, is around 5. So very highly infectious virus. It's the second leading cause of infant mortality worldwide. And we have 700 infants a day die from RSV, and they're largely in lower and middle income countries. RSV is also fascinating to study from the perspective of an epidemiologist. It has an epidemic seasonal circulation. So here in Wisconsin, like in Nashville, Tennessee, it circulates in the winter months, except that COVID totally screwed up circulation in the year, and we had a summer epidemic of RSV last year. But it circulates November to March in the Northern Hemisphere. And when it's our summer here, it's actually circulating in the Southern Hemisphere during their winter. And RSV doesn't have a non-human host that we know of. So we don't understand actually why RSV goes away in our summer months and circulates predominantly in winter months. It results in lower respiratory tract disease in about 20% of infants, and a really distinct lung pathology called bronchiolitis, also a common cause of wheezing in young children. And treatment for this, despite the fact that this virus has been recognized for over 60 years, is largely supportive. We used to have all these things we used to treat RSV, and once they were studied in randomized controlled trials, they were all deemed not to work. And so the reason these infants die in low and middle income countries is largely the lack of hospital level care and supportive care. We don't yet have an efficacious vaccine, although there are 50 right now in clinical trials. And there is RSV immunoprophylaxis, which is a monoclonal antibody, but it's licensed for a very small proportion of high risk, largely preterm infants or infants with chronic lung disease of prematurity. So next, I've told you about the disease we're going to talk about and the exposure that we're going to talk about. What's the link, historic link between these two things? Well, the link between RSV and asthma has been recognized at least as far back as 1959, when this first report came out from the University of Rochester, which has this long-standing history actually in RSV, dating to Karen Hall and others' work in this field, linking bronchiolitis in early life with asthma. And this was a case report of 100 children with bronchiolitis, followed up, of whom 32 went on to develop asthma. So this was first recognized in the late 1950s. This has been a consistently replicated significant association, by many groups, groups here, our group, among others. But this has left us with this decades-old debate and this continued study of severe RSV infection. And that is, does RSV cause asthma? Is there a shared genetic predisposition for both conditions? And that's why we see this association, or is it both? So the reason that we focus, among many other exposures, on RSV and asthma is because the population attributable fraction for RSV lower respiratory tract infection is higher than any other early life exposure. And what I show you here on this figure, these are the protective factors, these are risk factors, and this is RSV lower respiratory tract infection, a very high population attributable fraction, a relatively common exposure prevalence. But this is lower respiratory tract infection. And I'm going to tell you for the rest of the talk why we need to actually study RSV as an exposure, not as a clinical phenotype of disease. But this is why RSV has been so important in the field and why we focus on it as one of the potential early life preventable exposures that might decrease asthma prevalence. So the question you might ask then, if we've known about this for more than 60 years, is why have we not made more progress in understanding the causal relationship of RSV with asthma development? And I call this our Gordian Knot. And there's a number of reasons for this. The first is, as I said, since that first report, most all of our studies have focused on RSV lower respiratory tract infection as the exposure that we've studied. But RSV lower respiratory tract infection it's not an exposure. It's a clinical phenotype of infection severity. So it's analogous to studying cough with smoking as a risk factor for lung cancer. The exposure isn't the clinical phenotype that results from smoking. It's smoking that's the exposure. And the other point is that most of our studies of RSV lower respiratory tract infection and asthma are inherently flawed because the comparator groups don't define the presence or absence of RSV infection in the group, just the presence or absence of RSV lower respiratory tract infection. And then the other thing that we've tried to do, as have other groups, is use available agents like RSV immunoprophylaxis to study the effect on asthma development. And while we see an impact, the important thing to understand about these agents is, unlike something like measles vaccine, RSV immunoprophylaxis, and likely the RSV vaccines that will develop, don't result in sterilizing immunity. So they don't prevent RSV infection. They just prevent the severe sequelae of RSV, which is hospitalizations. So I'm going to talk first about this concept of shared heredity and asthma risk to explain why we can't continue to just study RSV, lower respiratory tract infections, to understand this causal relationship. So there have been identified a number of genes associated with severity of RSV, mostly candidate genes. And so there's really a small number of genetic studies of severe RSV. And there's only been one GWAS, and it's very small. It's 3 different populations combined with a total of only about 1,000 children. There are many studies of genes associated with asthma, including GWAS, and very well-established genetic risk factors. And this is then the shared genetic basis of this association that's seen in all these studies between RSV, lower respiratory tract infection, and asthma. But the problem is we don't know if it explains just a small proportion of the association that we see, or nearly all of the association that we see in these studies. So what we did is we used a meta-analytic approach, and we looked at all the RSV lower respiratory tract infection studies and asthma association studies. And we classified them as having no adjustment for any marker of a genetic influence. Largely these are family history compared to those with adjustment. And what you can see here are the estimates are smaller on average when adjusting for genetic confounding, which is the red. And in a very small study of monozygotic twins on RSV lower respiratory tract infection and asthma risk, you can see that the effect size, granted huge confidence intervals here, but is even smaller. So this is consistent with RSV lower respiratory tract infection, excuse me, at least partly being a marker of a genetic susceptibility for both diseases rather than purely a causal exposure. So let me lastly then explain why these studies ultimately can't get at the question of causality. So over here, and this might be too much Epi 101 for everyone, so I apologize for that. I'm going to stop after this slide. But over here on the left, A, this is where genes here are an unobserved confounder for the association between RSV LRTI, which is a very small proportion of all of RSV infection. In fact, hospitalizations for RSV, which is what a lot of the studies are focused on, are 1 to 3% of all infants. So I could have made this 1 to 3%, but then you wouldn't have been able to see the dot right there. And here, RSV LRTI is also a cause of asthma. In B over here, genes are an unobserved confounder for the association between RSV lower respiratory tract infection and asthma, but you can see there's no arrow here. And this is the model where RSV LRTI is not a cause of asthma. And the problem is, from these purely observational studies, we have no way to distinguish A from B. So we're never going to get at understanding the causal relationship with the model of these types of studies that we've done for the last 60 years. So how do we untie this Gordian knot? And if everyone knows the story of the Gordian knot attributed to Alexander the Great, it's where you have this intractable problem that's solved relatively easily by cutting this knot. And so I think of it really in a very similar way. The first is that we actually, as an environmental epidemiologist, we have to define something that's actually an exposure. And in this case, it probably would be being inoculated with RSV, but you all can imagine that no one in their right mind would design a randomized controlled trial where we would randomly infect some infants with RSV and not others, and then follow them to the development of asthma. So we have to study RSV as an exposure. And so we can't study inoculation, but we can study RSV infection, not a clinical phenotype of the disease. The second is that we have a way to measure our exposure. And the nice thing about studying RSV as a model of respiratory viral disease is we do have a way. We have molecular methods which are used in standard practice, so PCR. We also have RSV serology. So in infants who have asymptomatic or paucisymptomatic infection, at 1 year, we can measure serology for RSV to know if they've been infected in the first year of life. And then we can use birth timing as an instrumental variable. The thing that's so interesting about RSV from an epidemiologic perspective is this epidemic seasonality. So RSV we can use as an instrumental variable because it's a marker of RSV infection risk depending on when you're born. And then lastly, we really are stuck with these observational longitudinal studies, along with doing translational studies with our basic colleagues on the biospecimens we collect from these children to understand what happens to those who are infected in early life compared to those who are uninfected. So we've used birth timing as a random event. And we'd shown previously that when you're born, related to when RSV circulates, determines your risk for having lower respiratory tract infection with RSV. In fact, it predicts very nicely if you're at increased risk of winding up in the University of Wisconsin pediatric ICU with RSV. In a non-causal model, there should be no relationship to birth timing related to when RSV circulates on the development of asthma. It should just be a straight line. And in a causal model, and this is the very simple question we asked, which is there a relationship between asthma risk and birth timing related to RSV circulation? And if there is, what we would see is instead of this straight line, we'd see a very similar relationship to that of bronchiolitis. So that's what we did. We asked this relatively simple question about risk of asthma related to birth timing when RSV circulates. And we studied 95,000 infants over 6 RSV seasons. And we did 6 RSV seasons because RSV season shifts by, over this 6 years, it shifted by 8 weeks across these 6 different years. And we showed that the risk of asthma is associated with birth timing in relationship to RSV seasonality. Infants who are born at about 4 months of age before the peak of the RSV epidemic are at about a 30% increased risk of developing asthma. And this is an association that's really difficult to explain through non-causal mechanisms. And then we designed a population-based study of term, non-low birth weight, otherwise completely healthy infants, which started in 2013. We recruited through 2 years, and we identified in every single infant enrolled into this study whether they were infected or uninfected in the first year of life with RSV. And we did this through biweekly surveillance, using symptom surveys and PCR to identify RSV. And then at 1 year of life, all of these infants had RSV serology. And those who either had a positive PCR and or positive serology were deemed to be infected with RSV in the first year of life. And what we learned is that 54% of RSV infants were infected with RSV, 46% were uninfected. In fact, we had a larger proportion than we even anticipated who had relatively asymptomatic or paucisymptomatic infection. And this allowed us an assessment of exposure, yes, no, that's not confounded by this shared genetic risk. And in a normal term, non-low birth weight, otherwise healthy population of infants. But we asked still a very similar question. We were interested in whether, is there a genetic susceptibility to just being infected with RSV? We thought it was unlikely. The R naught is high. We know everyone in the world pretty much is infected with this virus by age 2 to 3 years. But working with colleagues Dylan Lawless and Chris McKinnon, we did first a GWAS, and there were no individual SNPs identified. But much more likely, this would have been a number of different genes that had some small cumulative effect, but we looked at that also, and the narrow sense heritability was actually 0. And then we looked at candidate genes for asthma and severe RSV infection. And again, we saw no relationship. So no common variants contributed to infection. It wasn't explained by small cumulative genetic effects. And even the most likely a priori SNPs weren't associated with asthma risk. So not terribly surprising. This is what we expected that we might see. But nice to know we aren't dealing with at least an obvious signal of genetic confounding. So in INSPIRE, we enrolled nearly 1,950 families. As I told you before, we think of this as a natural quasi-randomization. We didn't inoculate this. This was just natural infection. 54% of infants were infected, 46% uninfected. And those who weren't infected in the first year of life had significantly decreased risk of recurrent wheeze at age 2, 3, and 4 years of age, and a 28% decreased risk of asthma at 5 years of age, to support the fact that missing RSV infection in the first year of life confers a protective effect on subsequent wheezing and asthma risk. So if RSV does cause asthma, then through what mechanisms? And I'm going to present briefly data in 3 areas. The first is how RSV infection alters the airway microbiome. The second is the effect on the developing immune system. And then third, the developing airway epithelium. So first, this is work done by one of our K awardees, Christian Rosas Salazar, looking at the effect of acute RSV infection on the airway microbiome. And when I say airway, when we're talking about infants, I'm talking about the nasal airway. We're obviously very limited when we study children or adults about actually going into the lung. Really can't do that in a population-based study. So here in the green, these are the healthy infants. The red are infants with RSV infection. Christian had previously shown that this isn't an effect that's just viral illness, because we've compared the effect of RSV to rhinovirus on the airway microbiome, and it's substantially different. So in infants infected with RSV, we see an increase in Streptococcus, Moraxella, and Haemophilus. And this is a pattern that's previously been identified to be associated with increased asthma risk. And it's also why we speculate that infants, maybe after early life, are at increased predisposition to develop things like bacterial otitis media. But there were also bacteria which were decreased with acute infection. And most notable was lactobacillus. And it actually, although it doesn't have as high relative mean abundance in the airway, it had the greatest change with acute infection. And then here we modeled the probability of a nasopharyngeal bacterial genus actually being selected into a model that distinguishes infants who go on to wheeze compared to those who don't go on to wheeze. And in fact, lactobacillus is the most predictive in the upper airway of infants who later go on to develop wheeze. And when you have an increased abundance of lactobacillus, you have about a 65% decreased risk of subsequently going on to develop wheeze. And lactobacillus is a founder species. Infants are colonized with this when they come through the vagina during vaginal delivery. And that's the first colonization with this founder's species early in life. So potentially also a modifiable risk factor. And we think why RSV then may contribute to wheeze and asthma development is through its acute change in lactobacillus abundance during infection. So next, let me turn to the effect of RSV infection on the developing immune system. So this is another one of our K awardees, Kadir Turi, who's interested in metabolomics and the effect on the developing immune system. And this is looking at the impact of RSV infection during acute infection on nasal cytokine responses. So we measured 53 different cytokines, chemokines, and growth factors in the airway during acute infection. And this is a heat map on the left, but I'm going to focus over here on this PCA plot on the right here. You can see that there are 2 very distinct immune response patterns to RSV infection in early life. This one in green is characterized by a type 2 high, type 17 high, antiviral low response. And it's this group that has over a 3-fold increased risk of going on to be recurrent wheezers. So while this tells us that there's a difference in the response to acute infection in early life with RSV that distinguishes recurrent wheezers, this doesn't really tell us if RSV causes a change in the developing immune system, or this is the same differential genetic predisposition to RSV. So what we did next is a priori designed nested case control study where we collected PBMCs from 2 to 3-year-old children in the study who were RSV infected and RSV uninfected in the first year of life. And we stimulated these with mock, a clinical isolate of RSV, and then a positive control Staphylococcus enterotoxin B. And we looked at the type 1 memory T cell responses to virus. So a transcription factor, T-bet, and then a set of intracellular cytokines. And this is work done by Tanya Cherkova and Larry Anderson's lab at Emory when she was a postdoctoral fellow. And this is a summary of the data. In blue, the RSV uninfected in the first year of life. In red, the RSV infected, again on memory T cell responses at age 2 to 3 years of life. And what it shows is that RSV infection in infancy is associated with a subsequent dampened type 1 T cell responses more than a year following infection with this virus. And so then the last piece of evidence to look at in terms of answering the question of how RSV may contribute to asthma development is looking at the effect on the airway epithelium. So this is data from a publicly available source called LungMap. This is actually lung tissue, and you can download the RNA-seq data from this publicly available resource. But we looked at this data because we wanted to show the reason that it's so important to study the airway epithelium in early life. And what this shows is a very strong age-related developmental trajectory of human airway epithelial development that really ends at about 16 to 18 months of life. It starts first with basal cells, followed by the development of the junctions and secretory subsets of the airway epithelium, and then lastly by these mature ciliated cells to support that actually studying the airway epithelium in early life really is what's important before a mature airway epithelium is actually developed. So here we looked at 15 male subjects in our cohort at 3 years of age who were wheezers and non-wheezers. And we look at males and females differently because we see a lot of sexual dimorphism early in life between males and females. And I'm showing males here. And in this PCA plot, the blue are non-wheezers, the pink are wheezers, and you can see very clear distinction in these children. And what we're looking at here is carbohydrate metabolism of these live airway epithelial cells. And when we look at by NADH production, these non-wheezers and wheezers, what we see are that wheezers very early in life have different airway epithelial cell metabolism, and they use non-glucose sources largely for metabolism. And then here, what we looked at is the same 3-year-olds, markers of glycolysis and TCA intermediate metabolism, again, broken down by the males in squares and the females who are shown the right of all of these plots in the triangles. The blue represent those who are RSV infected in the first year of life. Those in red, the RSV uninfected in the first year of life. The red are RSV infected in the first year of life. And the differences in these markers of glycolysis and TCA metabolism that's different, not only between males and females, but across the board with almost every marker we look at of glycolysis between those who were RSV infected in the first year of life compared to the RSV uninfected group. So then how do we think, and what is the model for how this common early life virus may contribute to asthma development? Well, RSV results in this alteration of the airway microbiome, which I showed you early on, with an increase in these pathogenic bacteria and a decrease in this founder species of lactobacillus. It also results in what we believe are durable alterations in airway epithelial cell metabolism. This results in what we think is a reprogramming, resulting in a dysfunctional barrier of the airway epithelium, which sets up this airway epithelium to be more predisposed to aeroallergen sensitization and other environmental toxins, such things as pollution, and really characterizes this different airway pathologically that we know we see in those with atopic disease and asthma. So then the last part of the talk I want to turn to are understanding host and viral interactions of acute and chronic asthma development. And the thing that's interesting and also challenging about studying a virus as an environmental exposure is that the virus also can have genetic variability. And this isn't surprising. We know we've heard about for years different strains of influenza. We know H1N1 causes variable pathogenicity. Everyone in the world certainly knows this related to the SARS-CoV-2 virus, and it's really not any different with RSV. So using a haplotype-like analysis, we identified an RSV gene variant that's associated with variable infection severity and also recurrent wheeze risk in this dose-dependent fashion. So this is comparing the wild-type virus to a single stop A4G and a 2-stop A4G. So 3 variants of this virus and a very nice dose-response relationship with disease severity, not only acute disease severity, but subsequent wheezing risk as well. And so Stacy Human and Annie Hotard, also working in Larry Anderson and Marty Moore's lab, actually engineered this virus. And that's why here it's called R. This is the engineered virus from the clinical isolate. And they took this virus and they put it into an animal model. And the very surprising thing about this virus, while we could never understand or show why it causes increased pathogenicity in humans, what was very interesting about this virus is we were able to repeatedly infect these animals who had immunity, supposedly, to this virus over and over again, compared with other clinical isolates. And the reason for this, we believe, is that this 2-stop A4G compared to the wild type results in marked lower F protein expression on the surface. And this is the 2-stop A4G. Again, this is the equivalent of like the spike protein of the SARS-CoV-2 virus. And we believe because there are less of these glycoproteins on the surface, the animal is less able to recognize upon repeat infection. This strain has become the dominant circulating strain now. There's only about 5% or wild type of circulating RSV strains since we first collected these samples. And we think the reason is this immune evasion. So the other thing that's very interesting about RSV is we've recognized really for decades that there are these infants who look like they have repeat infection. And what we've really never known is, are these repeat infections or just persistent infections? So in Inspire, we defined children who had their primary infection with RSV, and 15 days or more later, they had a repeat symptomatic infection. And these infections were anywhere from 15 days to 3 months. And then what we did is we sequenced in all of Inspire, all of the RSV isolates in the study. And what we found out is that when you look at repeat infections compared to single infections, they're not repeat infections, they're persistent. It's the identical virus that we find 15 days to 3 months later in these healthy infants. So very interesting that we see these persistent infections. And so while the number of these were too small to genetically ask the question about whether there's some genetic predisposition for these viruses to persist over a long period of time, what we were able to look at is basically what's a viral GWAS and identify RSV variants that are associated with these persistent infections in these children. And this virus actually caused milder infection in all of these infants. None of these viruses cause lower respiratory tract infection. So we wonder also if these types of viruses that can persist in the airway epithelium are more likely to drive chronic changes because they persist, or even whether these are the types of viruses that are our reservoir of RSV. When we don't understand where is RSV in the summer and why does it just come back in November, perhaps there are these persistent viruses in our population with environmental factors that allow them to reemerge. So then lastly, we've looked at gene-viral interactions. So as I mentioned earlier, in asthma, there are many genes that have been identified, but probably the most important locus is the 17q21 risk alleles for asthma. And here we looked at 7 risk alleles. This was these were genotyped by Carol Ober's lab at the University of Chicago. And Chris McKinnon analyzed these, looking at gene-viral interactions. And what we did here is take these loci, and 3 were significant. I'm showing you just one here. The 3 that were significant look almost identical to this. But what you can appreciate is with the low-risk asthma loci compared to the high-risk asthma loci, with the red being the RSV-infected infants, the black being the RSV uninfected infants, that you can appreciate, depending on the asthma risk loci and whether you're infected or not, markedly changes your risk for subsequently developing asthma. So what do we think this model looks like? How do host genetics and viral genetics interact with each other? And this is what we believe, why we see this marked variability. So here, I've told you we can have these variable host genotypes that predispose you to developing infection. We think there are probably important interactions with virus. And I showed you that we have these viral variants that not only result in differential acute disease severity, but also differential persistence in the airway epithelium. So you can imagine if you're uninfected in the first year of life, you might have really low risk of going on to develop asthma, maybe the background rate, which is about 10% in the US. Infected with a wild type and maybe having a risk allele, maybe about a 40% chance of going on. But you can imagine if you have a high risk asthma allele and you're infected with a risk strain, that you might have an extremely high risk of subsequently developing asthma. And we believe this is the model for explaining variable susceptibility to asthma development. So let me just summarize then how a common respiratory virus may cause asthma development. As I said, I've explained why we believe asthma is a preventable disease and why we have to study RSV as an exposure. And through studying it as an exposure rather than a clinical phenotype, we've showed that birth timing in relation to RSV circulation is associated with asthma risk. Also, that missing RSV infection in the first year of life is associated with about a 30% reduction in asthma risk. Asking how RSV may contribute to asthma development, infant RSV infection, we know, alters the airway microbiome. It may alter the developing type 1 and type 2 immune responses, and certainly memory immune responses to virus, and probably contributes to airway epithelial metabolic reprogramming of this developing airway epithelium during a critical susceptibility period. That both the host and viral genetic variations interact to contribute to variable susceptibility to asthma risk. And the implications of this are that preventing or delaying infant RSV infection in the first year of life may decrease subsequent asthma risk. So then, you know, I'm an epidemiologist and I have a public health bent. Where do I think we take this? So I'm going to use cardiovascular disease as an analogy because I love this study that was done in Finland. So in the 1970s in Finland, they had the highest cardiovascular disease mortality in the world. And it was recognized even at that time that there were a series of modifiable risk factors for cardiovascular disease. And they undertook this major public health campaign on a population level to implement these changes within the country. And over a 20-year period, they decreased cardiovascular mortality by between 50% to 94%. And I think this is a really nice analogy for asthma. And I think this is what the future holds for asthma prevention. And I say, this is the slide I want to show before I retire, or it's the slide I want one of my mentees to show before they retire, whatever time frame that we're on. But we have a number of modifiable risk factors. And it is not, I presented RSV today, it's not going to be one risk factor that we can modify to prevent disease development. This is going to be like cardiovascular disease. It is a disease that is genetically, that children are genetically predisposed to develop. And there are going to be a multitude of what really need to be public health campaign interventions to prevent disease. And we have many. I showed you RSV infection, which is a high population attributable fraction, but there are others. Early antibiotic exposure alters the microbiome. We know this. Probably in diet, omega-3 has some of the best signals. Maternal stress, dogs and farming, which the group here is very interested in and studying. I think like cardiovascular disease, we're going to make the biggest impact in the young population. This is a disease with onset in early life. I think we'll see the biggest effects in the first year of life, but probably interventions need to be pregnancy and early intervention. And we're going to make these changes on a population level. That's where we will see the biggest effect. Yes, we need to study these, but the greatest effects we'll see are similar to what Finland did, which is taking a public health campaign, modifying, which most of these are things that we want to modify on a population-based level for lots of different reasons, not just asthma prevention. And we will see decline in asthma prevalence, I believe. So I'd just like to end with acknowledging the truly amazing group that I have the opportunity to work with. I acknowledged many of them during this talk, including 3 current K awardees in our group, and then our collaborators at Emory, Northwestern, Pittsburgh, and EPFL. And I also want to acknowledge the group here led by Jim Gurn and others as part of the ECHO Consortium, which I call sort of the new national children's study, of which we're very fortunate to be a part of and be able to collaborate with the group here. And I'll end there, and I'm happy to take any questions. And thank you for bearing with me for my first in-person non-Zoom talk, and I really appreciate your attention.
Tina, that was an amazing presentation and a tour de force. And I'll speak for Bill that he must be felling in his seat. It's a great legacy. Thank you for endowing this lectureship. And thank you for Tina for an amazing talk. We will open it up for questions. And I will hand it off to Dr. Bussey.
Yeah, Tina, I certainly second what Lynn said. That was spectacular and very encouraging. The question is on the modifiable factors. Where do you think the greatest preventative effort is going to be? You mentioned microbiome and sort of getting the good bacteria. Do you think that this is going to be a high priority, or do you think modifying some of the inflammatory responses from the T1, T2 pathway will be as good?
I think it's going to take a multifaceted approach. I think that I talked about one risk factor today and tried to convince you that there's a causal relationship with that risk factor in asthma development. But I truly think it's going to take a multifaceted approach. And there's lots of reasons for these other things that you mentioned. Normalizing, having a normal microbiome, C-section versus vaginal delivery, decreasing early antibiotic exposure. These are all things we should do for lots of different reasons, not just asthma. So I think of them as sort of no-brainers. But the secondary effects that you're alluding to, which are these different types of inflammatory responses that result from some of these, absolutely could also be a target of prevention. But I really think it's going to be a multifaceted approach like we've done with cardiovascular disease, where we're going to see the greatest inroads. I don't have my reading glasses on. Let me.
Okay. Are there RSV
quickly?
nucleotide sequences found chronically in susceptible individuals between active infection?
We don't really know the answer to that because we don't have that kind of serial sampling in these children. In fact, the study I showed you wasn't designed to look at persistent infection. We just recognize that this is a question that's been bantered back and forth for a long time, that we see these infants who appear to have these like repeat infections, and we've never really known, are they repeat or persistent infections? So unfortunately, we don't know because we don't have that type of serial sampling.
Can I ask one question? So in terms of the microbiome, do you know, is the gut microbiome also different in these infants? And then the second, can we simply inoculate with lactobacillus and improve outcomes?
So great question. So yes, in fact, most of the data has focused largely on the gut microbiome. I mean, and the gut microbiome is very important in terms of development of the immune system. And so yes, some of these changes are absolutely seen in the gut microbiome. Lactobacillus supplementation has been studied in randomized controlled trials. There's not a signal for asthma, but probably also not adequately powered for asthma. In an animal model, though, in a randomized controlled trial in an animal model, there's a big effect on RSV severity and the resultant respiratory pathology that results in the animals who are lactobacillus supplemented actually through the nose, so changing the airway lactobacillus. So yes, very interesting. And one of the K awardees whose work I presented, actually his K award focuses around actually taking the vaginal microbiota, and it's a feasibility study in inoculating C-section born babies actually after birth.
Then a question from Andrew Davis. given the differences in incidence of asthma in the urban and rural populations, is there also a difference in RSV infection rates in those populations? Can that explain that?
Yeah, actually, it's the reverse, which we don't entirely understand. So it depends on the population that you look at. So we know, for instance, Bill and Jim and Rob Lemansky's COAST cohort, about 50% are RSV infected in the first year of life, 54% for us. In Eureka, it's over 60%. So there is more RSV infection. I should say Eureka is an inner city cohort. So there is more RSV infection, but in most of the world, when we look at severity of infection, there's more severe infection in rural areas. But there is the infection incidence is higher in urban areas, at least in the small number of populations that we've looked at through our ECHO consortium.
Hi, Tina, that was a fabulous talk. I have two questions, but I'll start with one for time. The changes in the nasal microbiome that you showed, I guess my question is, was that at the time of infection? And the question is like, does that change the microbial trajectory in the child, and how durable is it? Like, have you looked at serial time points, and does it persist? Because we know, like with antibiotic use, you can, for some people will get a temporary alteration and then they'll go back to normal. Some people, it alters their trajectory.
So this is a great marketing setup for a study Anne-Marie is leading, which is the CANOE study, of which we're both a part, and we're collecting serial respiratory samples in this study because that's exactly what we want to know. We want to know how durable is this change? And I can only speculate then at this point because no one else has samples like this, like we're going to have in CANOE actually. And as you know, we're looking at not only the longitudinal history of what happens, but the impact of antibiotics also in that longitudinal. But what I suspect we'll see is that infants, like we all have as humans, we have some capacity for normalizing things when we get a hit. We see that with things like cholera and other things. You wipe out your microbiome, but we can reconstitute it. But I think the greater the number of hits, the less likely a child will be to normalize their airway epithelium. This is speculation now of what we're going to see, but I think you take the child who has multiple viral hits, multiple antibiotic courses, which is what we do a lot of times when they have a viral illness. And these will be the children who are unable to reconstitute a normal airway epithelium.
Well, I want to thank Tina again for an amazing discussion. And I know there's still a few more questions. We're at the hour, so people are welcome to come up to the podium and talk with Tina. But I want to give her a big hand again. And thank you all for your attention. And we're looking forward to more hybrid grand rounds in the future. So thank you. Thank you.